Good evening, everyone, and welcome. Thank you so much for tuning in tonight. My name is Kate Bruns, and on behalf of Harvard Bookstore, the Harvard University Division of Science, and the Harvard Library, I am so pleased to welcome you to this evening's event with Walter Isaacson, discussing his latest book, The Codebreaker: Jennifer Doudna, Gene Editing, and the Future of the Human Race, in conversation tonight with David Liu. Tonight's lecture is a part of our Harvard Science Book Talk series, which brings the authors of recently published science-related literature to our Cambridge community and now very far beyond. Coming up next week, we will be hosting events with Ewan Angus Ashley and Siddhartha Mukherjee and Nobel Prize-winning biologist Paul Nurse. To learn more about Science Book Talks, you can visit the webpage harvard.com/science or sign up for the bookstore's email newsletter at harvard.com. We also have a YouTube page where you can watch previous talks that you might have missed, and I'm going to be posting these links in the Zoom chat in just a few minutes. This evening's event will conclude with some time for your questions. If you would like to ask our speakers something, please go to the Q&A chat button at the bottom of your screen, where you can submit a question. We're going to get through as many as time allows for. I would also like to say a tremendous thank you for your patronage during these strange virtual times. Your support makes this author series possible and ensures the future of a landmark independent bookstore. So thank you to our partners at Harvard University, and thank you to all of you for tuning in and showing up for our authors, indie bookselling, and especially for science. And finally, you might have experienced in virtual gatherings, technical issues can arise. And if they do, we're going to do our best to resolve them quickly. So thank you for your patience and your understanding. And now I am honored to introduce tonight's speakers. A man of many hats, Walter Isaacson is renowned for his best-selling biographies on Leonardo da Vinci, Steve Jobs, Albert Einstein, Benjamin Franklin, Henry Kissinger, and more. Currently a professor of history at Tulane University, he served as the editor of Time magazine, CEO and chairman of CNN, and CEO of the Aspen Institute. He also co-hosts the PBS show Amanpour and Company, is a contributor to CNBC, and hosts the podcast Trailblazers. The Times of London aptly calls Isaacson a Renaissance man. Joining him in conversation tonight is award-winning chemist and biologist David Liu. Dr. Liu is the director of the Merkin Institute of Transformative Technologies in Healthcare at the Broad Institute, and is the Thomas Dudley Caput Professor of the Natural Sciences and Professor of Chemistry and Chemical Biology at Harvard University. Tonight, they'll be discussing Walter's new biography, The Codebreaker, about the life of Jennifer Doudna, winner of the 2020 Nobel Prize in Chemistry for her pioneering work on CRISPR gene editing. The New York Journal of Books calls The Codebreaker enthralling and panoramic, and Doris Kearns Goodwin writes, "When a great biographer combines his own fascination with science and a superb narrative style, the result is magic." We are so honored to host them for this conversation tonight. So without further ado, Walter and David, the digital podium is yours.
Thank you so much, Kate. Great.
Thank you.
Yeah.
So it's a pleasure, and I have to admit this is sort of an unusual role for me. I normally am interviewed and not interviewing, so I might completely suck at this, but I was really fascinated to read Walter's book, and of course I've lived through a good chunk of the book as a scientist in the field. So I'm as excited as I think the people online to be able to ask Walter some questions. And the way that this event will work is there's about 35 or 40 minutes where we'll be having a conversation, and Walter hasn't seen my questions in advance. And then there will be about 20 minutes at the end where audience members can ask questions, which will be moderated through the question and answer feature of Zoom. So if you have a question, you can type it into the Q&A box, and then it will be moderated and hopefully will show up on my screen, and I can pick from those and we can go over them at the end. All right, so Walter, I guess maybe the most basic question, but one which is probably on the minds of those who have followed your other works is, why did you write this book?
Well, I was enthralled by the digital revolution, which I did, through Steve Jobs and then the innovators. But Steve Jobs told me at the end of, as he was, fighting cancer, he said the next big revolution is the life sciences revolution. He was talking about his own, tumors being sequenced and the personalized medicine. And I realized that, having done Einstein and the physics revolution of the first half of the 20th century, and then the digital revolution in the second half of the 20th century, starting with the sequencing of DNA and then the development of tools to edit DNA, this would be an even more consequential revolution. And, I'm a Boston person myself, went to school up there, come back a whole lot, love the Harvard bookstore. And I realized Kendall Square was becoming the new Silicon Valley, and partly because you're there with the Broad Institute and at Harvard. But so I just wanted a way to write about the life sciences revolution. I wanted to focus a bit on RNA, which, as you know, is a delightful and miracle molecule. And as I was doing it, the scientist in China, He Zhuangqi, ended up doing designer babies. And then coronavirus hit and RNA became a essential part of a couple of the vaccines we have. And Jennifer and Emmanuel won the Nobel Prize. And so as I was writing the book, I decided Jennifer was a good main character, but all of your friends in Cambridge were also very, very eager and cooperative and spent much time in their labs, including George Church and Feng Zhang, who are also heroes of this book.
And you have such a broad background writing about different aspects of science and different revolutions within science. What about gene editing in particular made you feel like this could be the basis of, as you put it, the next great revolution, perhaps even more consequential than the digital revolution?
Right. Well, as a gene editing specialist, you would feel it. But even if you read the Nobel Prize citation, it says that this will bring science into a whole new epoch. And what it is, more than almost anything, I think the average person can relate to the fact that, oh, after 3 billion years, one species on this planet has finally developed the talent and the temerity to edit its own genes and hack its own evolution. And so that's something that captures the imagination quite well. And I think it's a great entry into all of the things that are making molecules the new microchip, meaning we're programming molecules the way, with the 4-letter codes of the genetic codes, but we're doing that the way we did microchips. And so I kind of felt gene editing was probably the most interesting of the technologies, but it also allowed me to do things like vaccines, which is a large section of the book, and to do things such as the structure of RNA, which Jack Szostak and Jennifer Doudna did at Harvard with the self-replicating RNA that gets the really big, big questions that we average people ask ourselves, like, how did life begin on this planet? And so to me, it was a good narrative thread with Jennifer Doudna as being the central character, but really just one of a cast of characters, and gene editing being the thing I think people can just grasp and say, oh yeah, that's going to change things.
Right. I mean, one aspect of gene editing that I think helps explain why it's captured so much of the public's fascination is that it scratches two very fundamental itches, as I see it in the human condition, that we want to understand ourselves, of course. We want to understand the relationship between our genes, our futures, disease, medicines. And of course, the second itch is we want to improve our own condition. And we'll talk more in our dialogue about.
Species, by the way, people keep saying, wasn't this bad? I say, wait, every creature, large and small, has that itch.
Right, exactly. So, gene editing in some respects is really the convergence of these two, I would argue, characteristic aspects of what it is to be human. So,
Boy,
one of the most important landmarks in the field of gene editing, and arguably the piece of work that earned Emmanuel Charpentier and Jennifer Doudna the Nobel Prize in chemistry in 2020 was the famous Genetic Charpentier Doudna science paper in 2012. I checked just before this event started. It's been cited 10,858 times,
wow.
which means in the past 9 years, it's been cited about 3 times a day, which is a lot for a science paper. And so I thought maybe, even though I feel a little embarrassed doing this to you because I normally ask undergraduates this question, maybe you could explain to me or to the audience what the Doudna Charpentier 2012 science paper did.
What they did was take this system that bacteria have been using for more than 1 billion years, in which they fight off viruses. And people had been studying it since really 2000 or so, because bacteria have these weird clustered, repeated interspace sequences, which get dubbed CRISPR. And it turns out that that system takes a mugshot, takes a tiny snippet of the genetic material of a virus that attacks them. And as you know, the fight between bacteria and viruses is the most brutal fight happening on this planet. So bacteria develop this trick, which is take a little mugshot of the virus. And then if the virus attacks again, it has a guide that brings an enzyme and knows how to cut that attacking virus. It's pretty useful for those of us involved in a pandemic these days. What Jennifer Doudna and Emmanuel Charpentier and Martin Jennick and Christoph Schilinski and that whole team did is they figured out the exact components of this CRISPR-Cas9 system, which means the guide RNA and then the enzyme known as Cas9 that can cut. And as soon as they figured it out and did it in a test tube, it really became clear that we could re-engineer that system. We could adapt that system, and we could change the target so it wasn't just a bacteria fighting a virus, but we could change the targeting so that it would cut our own DNA. And so it became the idea was we can re-engineer this bacterial trick that they have and make it into a tool to edit genes. And that happens in 2012 with the publication of the science paper. And it's pretty clear that it would be a good gene editing tool. There have been others, that you've done, which is, have names like ZFNs and talons. But it was clear this could be a really good gene editing tool. And then in my book, there's this race, a pretty stiff competitive race among many labs around the world. And in those 6 months of that race, they have to show how it could actually be used in a human cell. And in that race, Feng Zhang at the Broad and George Church at Harvard kind of beat in a photo finish the Jennifer Doudna group. They all published in January 2013, along with 2 or 3 other papers from around the world, showing how this system that Doudna and Charpentier had discovered could be used in human cells.
Right. Yeah, it was a landmark paper that really got many of us deeply interested and excited about gene editing, because as you pointed out, CRISPR is at its heart just a way to cut DNA. It's molecular scissors. But what makes it so special is that there have been at least 3 major classes of molecular scissors that scientists have used to try to edit DNA. You mentioned the ZFNs, or the zinc finger nucleases, and the talons, or the tail nucleases. And those methods work, but they are harder to use. They are more expensive to use. They require more specialized knowledge to use than CRISPR-Cas9. So it's an amazing example, I think, of how making something easier to use, cheaper, more accessible, really made this massive difference in how democratized the technology became for use in the life sciences, for use in academia, for use in industry, et cetera. So that's really been, to to me, one of the great take homes, which I think was beautifully captured in your book, of how that difference, this wasn't the first molecular scissors we knew how to reprogram, but it made all the difference that the scissors were so easily reprogrammed and so easy to deploy that students in laboratories around the world could quickly adopt the technology.
Even I could do it. I went to Jennifer's lab, and in 2 days, a couple of graduate students taught me how. And you're right, nature is beautiful that way, that you can discover something that bacteria have been doing for 1 billion years and say, we can adapt that. And the other thing I think you said that's so important is it's easier to reprogram. And that kind of echoes with the messenger RNA vaccines, why they're so transformative is when the variants come along and the mutations come along and some new virus comes along, we're going to be able to reprogram things. And this is a game changer in the history of the human species and its battle against pathogens.
Absolutely. In fact, what the audience, those who aren't specialists in the field, may not realize is the reason that we have these two mRNA vaccines, the Pfizer and the Moderna vaccine, so much faster than has ever been generated before, is because the mRNA vaccines that were nearly identical to the ones that millions of Americans have now taken were previously developed against MERS, which is the Middle Eastern sort of version of SARS. And it was simply a matter of changing a few of the letters in that mRNA sequence to now match the coronavirus sequence that allowed the vaccines to be developed and deployed and tested and manufactured so quickly. And that's what's going to, I think, be our best line of defense against these new variants, which, as you just mentioned, are worrying in that some of them may be able to resist to some degree the antibodies that these vaccines generate. So I want to ask you, what are some of the meta lessons that you've distilled from writing biographies about these highly impactful figures like Steve Jobs, Leonardo da Vinci, Benjamin Franklin, now Jennifer Doudna? Are there lessons that you think, that if you were giving advice to students, to young academics, to interested citizen scientists who just want to contribute to or be a part of or stay on top of science, what would you tell them?
I guess the main lesson would be the beauty of basic curiosity. Everybody I've written about, from Steve Jobs, to Albert Einstein, to Leonardo da Vinci and Jennifer Doudna, had that childhood sense of wonder that we all had, and that those of us who don't become scientists, we sometimes lose it. I mean, Einstein, as a kid, gets a compass from his dad, and he's mesmerized about how the needle twitches and points north. Well, I remember getting a compass. I was kind of mesmerized, and then like, oh, a dead squirrel, and I'm on to something else. But he's spending, he's just totally curious. Nothing's touching that needle. Likewise, Leonardo da Vinci is, fascinated by why there's a spiral in shells, seashells, and snails. And whoa, that's something Jennifer Doudna, growing up in Hawaii, was fascinated by. The molecules of life and why do they happen? And sometimes simplistically, those of us who write about the history of science and technology take what's called, a model of innovation that's called the linear model that Vannevar Bush came up with after the war, which is that basic science leads to discoveries that lead to inventions that become useful technologies and can be commercialized. It often happens that way, and to some extent, CRISPR is an example of that, because pure basic curiosity is causing Francisco Mojica, as a graduate student in Spain, to say, why are my bacteria sequencing this way? And basic curiosity is causing Julian Banfield and Emmanuel Charpentier and Jennifer Doudna to figure that out. But it's actually, to me, even more beautiful because there's a dance, an interplay between basic curiosity and applied science. And in my book, there's the yogurt makers, who you know well, Rodolphe Barangu and Philippe Horvath, and they're working for Danisco. And they've got all the historical data about the DNA of bacteria. And so they're noticing how the CRISPR sequences change, and they realize it's because of wave after wave of virus. So they protect the multi-billion-dollar yogurt culture industry. I guess the short form is we should all just stay curious about the wonders of nature and pause, maybe once or twice or three times a day, like Ben Franklin did when he walked by the Charles River and was wondering why the ripples on the surface of the river were going in a different direction of the wind. These are things we normal people notice, but we forget to be curious about.
I think that's such a great answer. That just following your curiosity, not following something that your mom or your dad expect you to follow, or that some career track that you're on has sort of pointed you towards. But following your curiosity is a way that you can get out of bed every day and feel very fortunate to be working on the thing that you're working on. So
absolutely, you get out of bed every
great
day and you
advice.
see the blue sky. And we forget to wonder, well, why is it blue? But I looked at Leonardo's notebooks and he has, why is the sky blue? And he sprays different types of water and purified water in the air to see what, and then Einstein, same question in his notebook. And he does Lord Rayleigh's formula and figures it out. Not only should we each day remember to be curious about the ordinary things we see, but we also have to make sure we don't smash that out of our kids by saying, quit asking so many questions.
Right. So you mentioned the sort of photo finish, as you put it, in the early days of CRISPR, where there was a race to demonstrate whether or not CRISPR could function in human cells. And academic science in general is known as an intensely competitive profession and institution.
Mhm.
So my question to you is, do you think the stunning rate of advances in gene editing that have occurred over the past decade are because of the competitive atmosphere you documented in your book, or in spite of this competitive atmosphere?
Oh, I think competition's a good thing, and I think it spurs people to go faster. And Jennifer Doudna is a competitive person, and she pushed the Science Journal, to publish her thing as quickly as possible because she heard footsteps racing behind her. And likewise, she and Feng Zhang and George Church were all racing to do things. Um, and so I believe that competition is good. But in any field, it's a mix of competition and collaboration and building on each other. One of the questions I have, and I'm going to turn this back to you because you will win a Nobel Prize in Medicine someday for the advances you've done in gene editing post CRISPR. But
I wouldn't count
I
on
think
that, but thank you.
we'll do the over and under bet. I'm better in betting that than I am on the March Madness brackets on basketball. But scientists are motivated, just by the way, journalists are too, by everything from prizes to patents to who publishes first to recognition, and also just for the joy of figuring out nature. And in the end of my book, I say, yeah, they were racing for patents and prizes and publications. But when COVID struck, they really all focused on, we have a noble mission, which is helping humanity. I don't know the answer to the question, but you're in the field. Do you feel that prizes, and for that matter, patents, are a spur to innovation, or does it sometimes uh, make it harder to collaborate?
I'm skeptical that prizes are on the forefront of most scientists. It's, the prize process, whether it's the Nobel Prize or less prominent prizes, is often pretty opaque to people. And I think you can spend a lot of time unnecessarily, a lot of energy worrying about that, or you could just focus on getting out of bed every day and being really excited about the question that you're dying to answer. So my guess is that most scientists aren't motivated very much by the thought of winning a prize. The competitiveness is real. I think for better or for worse, academic scientists don't really prioritize patents, but they do prioritize trying to make the discovery, trying to publish the paper, to announce it to the world. Being first matters sometimes. I think there's a newer trend, which I think is healthier, where when you realize that there's another group in your field that's working on the same problem, you join forces and you agree to co-publish together, or you agree to collaborate if it's early enough that you can sort of join your forces and still leave enough pie for everybody to get credit, for the students to be able to advance their careers. So
That's
I've seen
what
that a lot in
Darwin
this.
and Wallace did 150 years ago when they
Yeah.
decided to co-publish the theory of evolution.
And I've seen a lot of that in this field, and it's maybe one of the only ways that we scientists in the gene editing field and our students can stay sort of sane. That we try to, remind each other constantly that even our biggest competition is in the end on the same team. We're all working to try to bring these technologies so that they can advance basic science and they can advance human medicine. And if we keep those interests up front, I think it's a better guide than sort of any prize or worrying about getting scooped, even though it does suck to get scooped. I can tell
Well,
you from firsthand
even us
experience.
journalists worry about getting scooped. So you're playing on a higher field with what you do. And maybe so you're so diplomatic, I'll delegate you to see if you can get Fong Zhang and Jennifer Doudna and Eric Lander and George Church all to shake hands and get rid of all the patent
Well,
battles.
they're both terrific friends, reasonable people, and I'm optimistic that will happen. Speaking of which,
I
why don't
do
you tell
think the
us?
coronavirus crisis kind of have edged people that way. People are now putting up things on preprint servers. They're making it open. They're saying you can build on this if you're going to use it to fight COVID. Feng Zhang did, brilliant work with the Sherlock system to detect it. And likewise, Mammoth is doing it. And so I hope it's reminding people, let's all collaborate here. We've got a bigger enemy that's invaded us.
Right. While we're on the subject of some of these central figures, tell us a little bit about Jennifer Doudna's scientific partner, Emmanuel Charpentier, who may not be as well known here in the US as Jennifer.
Well, I loved going over she's in Berlin now at the Max Planck Institute, and just hanging out with her and being in her lab, because I speak French pretty well and grew up, knowing French, but I never quite knew what the word charmant really meant until I saw Emmanuel, and she's the most French charming person I've ever seen. But she's a bit reserved, and she has a shell around her, unlike Jennifer Doudna, who is just so genuine and friendly and will emotionally connect with you. And you see it in Emmanuel's career. Every 2 or 3 years, she picks up her pipettes and moves to another research university and doesn't really make any commitments, sort of moves on. She's peripatetic. And in her own life, she has very few commitments, but she has that Mona Lisa type smile that makes her seem both amused and knowing about things. And she had an interesting relationship, with Jennifer Doudna, because Jennifer's so outgoing and easy, and they meet in Puerto Rico and they collaborate. And I think, it sort of feels mysterious sometimes how cool Emmanuel can be, even though she's, truly likes something or cares for people. And so that she has a bit of an aloofness that when we were having dinner in Berlin and going to an art museum together, I tried to pierce.
Successfully?
Not totally. But in the end of my book, as you may notice, I had detected a sense of coolness between the two of them. And I did what I sort of suggested you could do with our other friends. And I just said, let's get on a Zoom call together, the three of us, and we can all talk about what it all meant. And as soon as they got on the Zoom together, I hid my video because they were talking at a mile a minute. And, Jennifer was calling her Manu, which is a nickname Jennifer uses. And that there was sort of a warming up there. So I don't try to make this a personality thing, but I do think whether it's Einstein or Leonardo, that personalities are part of the creative process. So I try to, and I've talked to them both a lot since then, tried to capture a relationship, because we all have that when we form a team. I mean,
Right.
Wozniak and Steve Jobs had that, had to deal with it. Michelangelo and Leonardo, don't get me started, that was a problematic one. But, this notion of how do you form a partnership is an interesting concept. It's like two chemical molecules that have to bond.
So I think you got an A even without Harvard's great inflated standards. You got an A on the easy part of this exam. So now I'm going to get to the harder part.
All
So
right.
in our remaining time in our dialogue, I wanted to walk you through a series of gene editing applications. Some are very current and some are a ways in the future, but will probably happen, at least be a possibility in the lifetime of the vast majority of the people on this call. And I wanted you to share your thoughts on whether or not you consider each of these applications ethical. And they'll start maybe on the easier side, and then they'll get a little bit more complicated. Okay, so the first is an example of a monogenic, very serious genetic disease. That is a disease that greatly shortens somebody's life. It's terrible if you have the disease. It's caused by a single letter change, a disease like progeria or sickle cell anemia.
Or Huntington's, yeah, which is just
Or Huntington's.
gross.
There's many, cystic fibrosis, Tay-Sachs disease, Rett syndrome. What are your thoughts on using gene editing technologies, some of which have already been shown in animals to be able to rescue the disease, use them in human patients?
I'm totally in favor of it. And of course, it happened last year to a wonderful woman in Mississippi named Victoria Gray in my book, who had the stem cells of her blood edited so that she no longer was producing sickle cell and she could lead a normal life. And when sometimes I'm in these discussions, at first, I recoiled a bit at gene editing. Have we been like Prometheus and snatching fire from the gods? But like Jennifer Doudna, I would keep meeting people, including today. Somebody called me up and said, I've read your book and I've got a 12-year-old daughter and she's got one of these diseases. But they say she's going to die. Can you get me in touch with Jennifer Doudna? So the answer to your first question, not only do I think it would be moral to do those type of edits, it would be immoral not to do them.
Okay, second, so that was number one. Case number two, genetic deafness. Okay, so you have mutations in genes that cause hearing loss. Often it's a rapid progressive hearing loss that results in profound deafness. The patients are often otherwise completely fine.
Yeah.
And of course, some in the deaf community would argue that deafness is not a disability. In fact, it, in some people's opinion, opens up a whole rich culture that otherwise is inaccessible.
It's a
What
totally
about
wonderful
correcting
question.
genetic deafness?
Yeah, it's a totally wonderful question. As you know, I deal with that. I have a whole step by step as we deal with this in the book, because we all have to go down this slope. And slopes are a little bit less slippery if we do it the way you're doing now and the way I do in the book is okay, what about this next step? On deafness, it's particularly interesting because you can definitely argue that it is a true disability in any species. You're not going to be as abled if you're deaf, and it can provide dangers and everything else. But yes, I was sitting on the balcony right behind me while pondering this about a couple of years ago. And it was, there's a gay pride parade and there was, I don't know, many things happening, but there were Gallaudet University students all signing to each other. And so in my book, I talk about some people who convince or try to convince me that yes, the deaf community is adding something very interesting to our species. And we don't want to edit out the diversity that you can see walking down the French Quarter on Royal Street and say, okay, let's edit deafness out. However, if you ask me, and I were in a fertility clinic, and it said, we're not talking about the whole human species, but you have a child who has this gene for deafness, or a fertilized eggs, but we can edit it so the child won't be deaf. I would choose not to have my child deaf, but I sure have had my mind open to the fact that it's partly a societal problem that we don't accommodate that diversity, meaning deafness, and we hearing enabled people, consider it just a disability instead of part of the great fabric of humanity.
Right. That's I think, a very thoughtful way to put it. All right. So examples 3 and 4 are both Alzheimer's disease.
Yeah.
So example 3 is an allele called ApoE. So this is a gene variant that most people probably have 2 copies of ApoE3. A small fraction of the audience has 2 copies of ApoE4. If you have 2 copies of the ApoE4 variant, which is just 2 one-letter differences between the ApoE3, your Alzheimer's risk is about 15 times higher, which is a very substantial change. And a small fraction of the audience might be ApoE2, which is 2-letter changes in the other direction. Those people actually have a substantially lower than normal chance of getting Alzheimer's. So what about taking an E4 person and editing them to E3? That might be more like the first examples that we talked about, although it's disease risk, disease prevention, not quite 100% chance that you're going to get Alzheimer's, even if you're ApoE4. And then, of course, what about turning people who represent the vast majority of the audience, who are probably ApoE3, editing them to become ApoE2, lowering their Alzheimer's risk?
Well, my mother-in-law has Alzheimer's now. A friend of mine, somebody you actually know who's been involved with Harvard, has it. And yes, I would make those edits. And one of the moral questions I would ask is, if you go, say, from the 4 to the 3 or the 4 to the 2, are you creating something totally new, or are you just doing something that in the wild, as we call it, a large number of our fellow members of our species have? And if you're doing something that is species typical or is widespread in the species, and it means you're going to lead a much more productive life, yeah, I would definitely go there.
So it's a great segue to case number 4, which is also Alzheimer's disease. So 0.1% of Icelandic people, and virtually nobody else, has a rare mutation in a gene called amyloid precursor protein. It's called the A673T mutation, single letter mutation. Gives them a much lower than normal chance of getting Alzheimer's. So should we install that mutation? Or another way to look at it is the Icelandic 0.1% of Icelandic people have the normal gene, the less Alzheimer's disease prone. The 99.9+% of the rest of us have the disease prone form. Why not install that mutation in people?
I would be in favor of that. The obvious thing that we have to just do as a caveat is you have to make sure there's no unintended consequences, that it's totally safe. But I assume for this thought experiment, feel like I'm in Michael Sandel's class or something, but the thought experiment we're doing is, once we know it's safe, I don't see why we would not do that. I don't know if you're going to go to the next part, is would I enhance memory, but that's where I think we have to draw a blurry line, which is, should we prevent bad, debilitating memory loss? Yeah. Enhancing
Okay,
memory?
and the final example, and then we'll go to the questions from the audience. So at the 1964 Winter Olympics, a man named Eero Mantaranta won the cross-country skiing competition by a margin that has never since been equaled. He also then won a lawn skiing competition by another sort of absurdly large margin. He was a Finnish skier, and it was later found by sequencing his family members that he has a single mutation in his erythropoietin receptor gene, which basically means he is always on EPO. It is as if he were doping with the Lance Armstrong EPO scandal drug, except it's natural. He can't help it. His EPO receptor is always sensing the presence of EPO, even though he's not injecting it. And that's what accounts for his incredible cross-country skiing ability. So what if people say, I occasionally am out of breath. I have, I would find it very useful to have longer endurance. Why don't you make that same mutation? It's a single G to A mutation in one position of this EPOR gene. How would you feel about using gene editing to install that mutation in people?
Well, that's in my book too, as well as is he. And of course, as you know, they accused him of doping. He had to prove he wasn't really doping, and they found this genetic quirk that he had. I think that crosses a line, to me at least, which is this is not medically necessary. This is just a pure enhancement if everybody wants to have the endurance, of this great cross-country skier. And I think it would sort of spoil athletics. We'd go from admiring the athlete, and we'd have to go instead to admire his genetic editor who gave him the genes. But, on the other hand, some people in the natural lottery, they get this. Most marathon runners have, a single gene mutation that helps their blood have more oxygen. Why is it more fair that they got it by the natural lottery than if they got it because their parents paid a really good genetic engineer to make sure that their kids had better genes for uh, endurance? So I guess my answer, and Michael Sandel's, who I consult with in this book, is there's something we should be reverential about the natural lottery and what we are gifted. And sometimes it's not a gift, if it's Huntington's, if it's Alzheimer's or whatever. But we should also respect that our species, our empathy, our morality partly comes from the fact that we're all subject to a bit of a random lottery in the genes we get. And I would not want to cross lines that let rich people enhance the sporting ability of their kids, and we were not all one species and one humanity.
Right. And I think that line is not a line, it's more a graded shadow.
But as
And
Leonardo da Vinci taught us, sfumato can make distinctions, even if it's not a sharp line.
right. And to be clear for the audience who isn't mostly in this field, there are many, many technical challenges that currently make most of what I just described virtually impossible right now. Although you could imagine within 10 years, some of those types of edits might be possible.
I think I just
All right.
noticed the athletic director of Harvard who was on this sign off and go run to see if he could find this technique.
Okay, so questions from the audience. So Todd Ryan asks a wonderful question. He writes, I'm a high school biology teacher. One of the things I desperately want to do is to motivate my young female students to get involved in science. How will your book facilitate that for my students?
Well, I hope it will. It's one of the many reasons I wrote it, because Jennifer told me that when she was in sixth grade, actually a little younger than her students, she came home one day and found that her dad had left a paperback copy of the double helix on her bed. And she thought it was, some detective story she put aside. But when she finally read it, she realized it actually was a detective story about the secrets of life. And she kept noticing this character, Rosalind Franklin, who Jim Watson treats rather condescendingly. But Jennifer told me, I didn't notice the condescension. I noticed a girl could be a scientist. And so she went to her school guidance counselor, said she wanted to be a scientist. And this is in Ilo, Hawaii. And the counselor said, no, girls don't become scientists. Well, she persisted and she did. And so part of my hope for this book is that if people do read it, they may say, okay, now that I'm finished, let me leave it on the bed for my daughter or my niece, or for that matter, my nephew or my son, because if you got role models, you'll be inspired to do something really noble. And that's what scientific research is. It's an endeavor that's really noble.
Right. So Terry Shepard, who I know and is editor of Nature Chemical Biology, a very important journal in the field of chemical biology, has a really, I think, important comment. He said, the potential for human gene editing is a great hook for public interest in CRISPR, but many non-scientists may not know how much CRISPR has revolutionized basic and biomedical science and how it enables discovery. And could we comment on that? And I think, Terry is absolutely right here. In fact, Adgene is sort of the nonprofit library of DNA blueprints. And every time Adgene sends to a scientist who requested one of our blueprints, that sends that blueprint, they let me know. And so Adrian has told me that 13,000 of our gene editing constructs have been sent out to thousands of laboratories around the world. And the vast majority of those requests and those people are using it for basic science, are using these tools to make changes precisely in the sequence of DNA of an organism or of a cell, and looking at the effects, the consequences of that change.
Yeah, it was like RNA interference was for a long time and is still, which is, oh yeah, you can look at the consequences of something in basic science. But it gives me the opportunity, because you've said that you're used to answering questions and not asking them. And I'm used to asking questions and as opposed to answering them. And I want to turn it to you, which is not exactly what that question was, but the frontier of gene editing. CRISPR took it a quantum leap into a new orbit, but now it's still progressing. You're on the forefront of it with prime editing and base editing. Do you think things are moving really fast now in making accurate tools, not only to cut DNA, but to put in replacement genes?
Yeah, so that's a great question, and it actually overlaps with one of the questions from the audience, from Linda, which was basically how can sort of cutting DNA actually fix things? And so as you pointed out, CRISPR is scissors that cut DNA, and just like the viruses use the scissors to destroy bacteria use the scissors to destroy a viral gene. If you cut the gene encoding diseased hemoglobin, you don't fix the hemoglobin gene, you actually mess it up further. So there are some diseases, sickle cell anemia being one, where you can cut other genes that, for example, turn off a redundant kind of hemoglobin that, if not silenced, would actually help those patients. And that's what Victoria Gray has had done to her, some of her blood stem cells. But the two technologies you mentioned, base editing and prime editing, came out of our lab's desire to use these gene editing technologies in ways that directly fix mutations that cause disease, instead of cutting genes to disrupt them. And so base editing is a way that you can change one letter of DNA to another letter. We can do it 4 different ways. We can change A's to G's, G's to A's, T's to C's, or C's to T's. And then prime editing is a newer technology where, like base editing, it doesn't cut the double helix, it doesn't disrupt genes. Instead, it makes virtually any local change, any small insertion, any small deletion, any base-to-base conversion. And each of those technologies, along with the CRISPR scissors, has its own strengths and weaknesses. They've all been widely used by the community to do different things. And I'm optimistic that each of them will also have a role in advancing this sort of new era of not being beholden to mistakes in our genomes that affect roughly 1 in 10 people with a grievous genetic disease of some type. So I was just going to say, so I appreciate you bringing up that topic, but I think the field of gene editing continues to advance, and just as it has built on this programmable way of cutting DNA, now scientists are using non-cutting ways to directly fix mutations like base editing and prime editing, and in the future there will be other ways. There's labs, including Fan Zhan's lab and Sam Sternberg's lab at Columbia, that are working on these wonderful CRISPR transposases that can insert large pieces of DNA, so far not in human cells, in bacteria, but that might also be a new capability that could revolutionize what people used to call gene therapy, introducing healthy copies of genes where they are defective or missing in patients. As well as, of course, studying biology.
The transposons one is one of my favorite of the new leaps that you all are doing, because it's really a jumping gene. And it was first discovered by another great woman, Barbara McClintock, at Cold Spring Harbor. And I remember being at Cold Spring Harbor with you and Sam and others when that was coming to the fore. Because it used to be, to answer the question directly that was asked, we had thought if you made the cut, you could have what's called homology-directed repair, or something like that. George Church had figured out templates. It doesn't actually work that well, and that's why what David is doing and what Sam is doing and what Fong is doing, it just astonishes me. I'm glad I got the book out because this field is racing ahead. I'm gonna have to do a addendum for the paperback.
So another Linda asks a question, which I think is a wonderful one. If scientists put an edited gene into a creature, um, uh, do the edits spread to all the other cells in the creature? How about to the creature's progeny? Maybe you could comment on that and how it relates to Dr. Hu's experiment.
Yeah, that was the big line that Dr. He and China crossed, which is no, if you take the stem cells of Victoria Gray and make an edit, or you do it in the eye cells or other things, it doesn't spread to every cell in the body. And that's the safer, that's the way we're all kind of comfortable. It's called somatic editing. You can cross a line, and it's called the germ line, which is you edit in a reproductive cell or very early stage embryos, and that means the edit goes to every cell in the body. And if it does that, it means it goes to reproductive cells in the body, the cells that produce sperm and eggs. And so it means that those type of edits done in these early stage embryos or sperm and egg not only applied to the person that's being edited, but to their children and all of their descendants. And that's where I think we have to take a deep breath, but even though the Chinese scientists did that, before we go barreling past the germline.
Right, and I think one of the most problematic aspects of Dr. Hu's, what's now affectionately called the CRISPR baby fiasco, is that there really was no medical benefit to these little girls. Because even if they had the edit that Dr. He claimed that they had, and there's some reason to believe that they actually had a mixture of those edits, in one case, it wouldn't have even offered any protection against HIV infection. And in the other case, there's now a messed up copy from these scissors of a gene called CCR5 in this little girl, and we have no idea what that messed up protein is going to do. So it was a real wake-up call to the field for all sorts of reasons, well documented in your book. Susan asks, is there a Rosalind Franklin equivalent in the gene editing space? It's a great question. Maybe you can first explain what she means by Rosalind Franklin equivalent.
Well, I think I would answer to some extent Jennifer Doudna. Rosalind Franklin was a very good structural biologist. And one of the key clues in the double helix and my book or whatever is that to understand a molecule, and David and I were talking before this about the spike protein even, and this molecular thing, you have to understand the shape of it. It's not just the chemical makeup of a molecule, but its twists and its folds. Jennifer was a great structural biologist. She used crystallography and other techniques in order early on at Harvard as a graduate student or postgraduate student to do the shape and twists and folds of the type of RNA. What Rosalind Franklin did was the crystallography images that were used without her permission by James Watson and Francis Crick and Maurice Wilkins to study the structure of DNA. I think there are a lot of women in the life sciences now, obviously Emmanuel and Jennifer and Jillian Banfield, but what's particularly heartening is that 60% of the people studying biology in college or graduate school are now women. So I think this will be a more inclusive revolution.
And I assume what the questioner meant by the Rosalind Franklin of gene editing is also not just a reference, as you, I think, wisely pointed out, that Rosalind Franklin and Jennifer Doudn were both structural biologists, but Rosalind Franklin is widely considered to be maybe the most important contributor to the solving of the double helical structure of DNA that at the time at least did not receive her due appropriate recognition. So, you feature a number of people, the vast majority of which probably won't be well known to the audience, at least until they've read your book. Are there people whose contributions to the field you can highlight as, perhaps deserving of more recognition than they've been given thus far?
Well, when Jennifer and Emmanuel won the Nobel Prize, I think I mentioned in the book, I could sense a tight smile on the face of Rosalind Franklin up in heaven as she's looking down on it. but yeah, I mean, I'm going to sound really craven here, but you're one of them. And that's why I wanted you to be the moderator here. I think that you and Fang Zhang, and to some extent George Church, but he's been in the field a lot longer than you have. I would say you and Fang Zhang and the Broad Institute, the people in that orbit, are the wave of people coming along that aren't well known. But
Well,
can I turn
I'm
the
embarrassed now because I wasn't, I didn't mean, I didn't mean that.
Oh, okay.
I meant instead sort of in the early days of CRISPR, which
Yeah.
CRISPR basic biology, which I definitely was not a part of. And most of the people you just mentioned weren't a part of.
Yeah. Well, I guess Francisco Mojica, is that a
Yep,
good answer? So
that's one.
Francisco
I think
Mojica
Burgess Sixness is
and
another good
Virgin
answer.
Sixness
Yep,
and the two yogurt people I mentioned.
yep.
Francisco is one of my favorites because he was a graduate student in Spain, in Alchiante, Spain. And he keeps in the late 1990s noticing all these sequences in bacteria. And he thinks he's screwing it up because gene sequencing was not very easy back then. And but he keeps persisting. They say, well, don't worry about these repeated sequences. He keeps persisting and persisting. And not only does he finally figure out what they do, he's the one who coins the name CRISPR. He's also just a total delight to meet and talk to. So his pictures in the book is one of the early heroes. Virginus Sixnius is a very shy Lithuanian, but he was the one racing with Jennifer Doudna and Emmanuel in 2012. And he had almost all the elements of the CRISPR system at around the same time they did. I won't get into total details, but there's something called the tracer RNA that I don't think he had exactly right. But he's still a hero of mine in the book as well.
Right. And as I understand it, he actually submitted his paper describing many of those key results before anybody else, but the paper was rejected from the initial journal that he submitted it to.
Well, not only, I think 4 journals. I mean, it shows that if it's better to be near Kendall Square when you want to get published than in Lithuania, because people are saying, what's this paper from Lithuania trying to describe this? So yes, I try to give him his due, and I'll put his picture in the book too.
So Lily asks, how do you, and a number of other audience members ask questions along the lines of this one, how do you think we can better justify the costs, which would likely be exorbitant, of using gene editing in sort of small N of 1, N of 5 clinical trials for rare diseases to the general public? In other words, if I can rephrase, these treatments and developing these treatments are going to be very expensive. How can we maximize the access that we all of us working in this field want society to have so that as many people as possible benefit from these technologies?
It's a really good question. And after He Zhongqi did did the CRISPR babies, a fiasco as you call it, in China, Jennifer and George Church were called in front of the Senate committee to sort of testify, have a hearing on what this all meant. And very early in the hearing, people quit asking about the Chinese babies, and they started saying this question that you just asked me, which is, these seem like miracle technologies, but how are we going to pay for it? Is it going to break the bank? Victoria Gray's treatment for sickle cell probably cost $1.5 million. What Jennifer has done is taken a scientist I assume you know pretty well because he's an amusing person, named Fyodor Urnov. And at the IGI, the Genetics Institute that Jennifer has been director, gave him the task of saying, how are we going to bring down the cost dramatically? Part of it is not in the CRISPR gene editing part. That's not the cost. The cost is harvesting stem cells and then, putting them back in the body. That's where the real cost is. So the question is, can we develop treatments where you don't have to take out the stem cells? Can you do it in the patient? And so those are ways to do it. But here's the ethical tricky thing. If you really want to save money, you do it in early stage embryos and you make it inheritable. That's pretty cheap.
Which is called clinical germline editing and is one of the red lines that very few scientists are willing to cross.
Right. But if you really wanted to make it very inexpensive to take on the millions of people who have sickle cell, you'd say, okay, let's do germline editing, make it inheritable and do it in early stages. And it's what the Chinese scientists, he crossed that line and got, in jail.
And
But
in part, that's a tough line to cross because the patient cannot consent to the treatment, right? No embryo can give informed consent to the treatment, and therefore no embryo can evaluate thoughtfully the risks and the potential benefits of the treatment. And of course,
we
all
do
of that
not.
embryo's progeny, if they reach childbearing age and choose to reproduce or are able to reproduce. So it's an incredibly ethically fraught question, and one reason why so many scientists, including myself, are opposed to clinical germline editing at this point.
There's a wonderful bioethicist in my book named David Sanchez. He happens to only be 17 years old, and he has sickle cell. And your friend Matthew Porteous has been treating him, and Matthew says to him in the book, I quote him, as, we're able to treat you, but we could also someday do edit, so your children and their children will never have sickle cell. And David says, wow, that would be wonderful. But then he pauses and asks that question that you just did, because he's a smart 17-year-old. And he says, but I think that should be up to the kid after the kid is born. And they say, why? He said, well, I didn't like having sickle cell, but it taught me endurance and patience and persistence. It forged my character. It taught me empathy for other people. So I'm not sure I want you to edit my children, without them consenting. But,
Right.
it's tricky. I asked him a few months later, I said, would you really not want that? He said, no, I've thought about it. I don't want my kids to have sickle cell. I double over in pain every time I play basketball. So I guess I would have them edited. So there's no, I'm going to destroy sales of the book. There's no answers at the end of the book, but we get to meet people like David Sanchez who try
Well,
to
that's why we
mark
know it's a good book, because you don't try to
Yeah.
write superficial answers to these very non-superficial questions.
Mhm.
So an anonymous attendee at 7:48 p.m. For the Harvard Bookstore moderators who are taking the questions off the list, says, hi, David, Walter, how do you think the CRISPR world should be presented to the general public so there are no miscommunications and concerns that impacted agriculture in GMO, which results in lots and lots of people still feeling like GMO crops are not something that they can they can feed their families with?
Well, once again, it's another reason I wanted to write this book, because it's fine with me if you've got strong opinions about GMOs. However, I think it would be useful if you knew what a gene was and what a genetically modified organism is and what it does. And to me, as a storyteller, the best way to make people understand science, be less frightened by it, to demystify it, is to make it a personal journey of discovery. So that's the narrative thread of my book, which is watch these scientists get to know the fact that they're real people who grab their kid from a robot building camp when COVID strikes and then turns their attention to, fighting COVID. And don't be anti-science. Watch as they wrestle with these moral issues. I think there are hundreds of ways we can communicate science better. This is being done by the Harvard Bookstore series like this of communicating science. My way of doing it is by doing a narrative journey of discovery with real people in it. And I hope that demystifies the process.
So Hans-Peter Kim, who is a world-class biologist.
I know the name.
Okay, he has asked a question. Great book and conversation and discussion, especially regarding germline editing and potential ethical implications. Could you comment on how genetic modifications of disease risks, like the Alzheimer's example, could or should be regulated? Who should decide which disease risks are okay to consider for gene editing or modification? And there's a whole cluster of questions that are related to the topic that Hans-Peter just described.
Yeah, I think it should be a societal discussion. I don't think it should be delegated to, experts or whatever. And so we should decide, and by we, I mean everybody on this Zoom call, everybody reads the book. That's part of the process of why don't we help think that through as a society? It has to be done internationally, and that's why Jennifer organized with David Baltimore and many others, and Robin Lovell Badge from England, and Duangqing Pei, somebody I spend time with who's in the Chinese Academy, because we're going to have some international consensus on this as well. And the real question that gets you back to whether you took John Rawls's course in justice or Michael Sandel's course in justice is, should it be decided by the individuals or should the society have rules? For example, if you let it be just a free market individual choice, like we do with many of our reproductive decision choices, then you can imagine a society in which, people like me would say, no, I want to edit out the gene for deafness, or I want to enhance the genes for endurance or memory. And rich people may be able to pay to do that. And you would have a eugenics that's not a state-sponsored eugenics, but is a free market libertarian eugenics. That's one way to do it, is let individuals make that choice, which is very much an American sort of instinct, is let it be up to the individual. The other is that society should have some say in these broader issues. So that's a core ethical question. When you say who should decide, should we leave it to individuals, or should there be an international and social consensus enforced by the FDAs around the world to say, we're not crossing this line, we're not using it to get rid of deafness even. So that's
Right.
explored in the book with the help of Michael Sandel.
All right, I think we're over time, but I want to end with one question that Randall asks, which is, Walter, what book project did you put aside when you started working on Codebreakers, and what book is coming out next?
I don't know what's coming out next, although I'm kicking around. I put aside a book on Louis Armstrong. If you look behind me, as I said, you'll see neighborhood where he grew up. I was interested in race and New Orleans and the 1900s, when we didn't have a color line here in New Orleans. They hadn't drawn the color line at the time, meaning there wasn't a big distinction between Creoles and Octoroons and stuff. But in the 1890s, we did that, and jazz emerges out of that. But I learned everything I could possibly know about Louis Armstrong, what he did every single day, but I couldn't crack the code, to use the phrase code breaker. I couldn't figure out what he was thinking, really, whether he was happy, whether he liked white people. So sometimes you do a project and you're not the code breaker. You can't crack the code on it. So I put it aside, but someday I'm going to persuade Wynton Marsalis to co-author it with me. And I'm going to try to crack the code of that, because I do think creativity and diversities in societies are an important thing to understand.
All right, well, I think we'll bring our dialogue part of it and the question and answer to an end, and I'll turn it back over to Kate if you have any closing remarks.
Yeah, I just wanted to say thank you tremendously to both of you for this relevant and fascinating discussion. And thank you to everyone for joining us tonight. If you would like to read more about CRISPR and Jennifer Doudna, copies of Codebreaker are for sale on harvard.com. But that's all that I have. So I guess I'll just close us out. On behalf of Harvard Bookstore, the Harvard Division of Science, and the Harvard Library, have a good night, everyone. Please keep reading and be well. Thank you.
All right, take care, everybody.
Thank you.
Or in the immortal words of Fyodor Urnov, stay positive, test negative.
I get to interview David next time. We do Home and Away.